Welcome, student. In this sessions, we are going to discuss the alternative to practical exam solutions. IGCSE Biology Paper 6 Alternative to Practical Exams consist of the following main parts. Number 1, data calculation and analysis, plotting the graph, the drawing the diagram and magnifications, and planning and investigations. In these sessions, we are going to discuss the data calculation and analysis. Now, first question in the paper 6 exams always will be investigation. And in this investigations question, you're supposed to read the investigation carefully and rephrase it. For example, a student investigate the effect of two different types of washing powder on the stained clothes. One was biological and contained enzymes and detergent, while the other was non-biological washing powder without the enzymes. So you're supposed to rephrase it. So in the rephrasing, in your own work, like effect of the different washing powder on the stained clothes. And further, when you're reading the investigations, the following point you're supposed to keep in mind and you're supposed to underline during the reading. For example, the point to underline include number 1, dependent and independent variables. Number 2, the variables that remain the constant and the same. And number 3, the controlled variable. And number 4, the potential source of error. So these 4 points you're supposed to underline during this investigation. For example, in this investigations, they were provided with the 4 pieces of a dry clothes that had been stained with the same food. Now here, the same food that is are the constant variable. Second, a solution of biological washing powder. So there's a one variable which is changing, that is a solution of biological washing powder. There's another variable, a solution of non-biological washing powder is also a changing variable. While the distilled water in which there's no detergent, that is the control. So these are the points during the reading you're supposed to underline. Further, we are moving toward the different step. You're supposed to read it carefully. The step number 1, a marker and the pen was used to divide the white tiles into the 4 sections. These 4 sections were labeled the bio and non-bio and water and not washed. One piece of the stained dry clothes was placed on the not washed sections while white tile. While in the step number 3, one piece of the stained dry cloth was placed into each of the solutions that labeled bio and non-bio and water, and one glass rod is used to place each cloth of bio and non-bio and the water into the water beaker and stirred for 10 seconds. Now here, one glass rod is being used to transfer the cloth into the different solutions. So in this way, there is the source of contamination. So there is potential source of error. So you're supposed to identify that. And second, the time duration is a constant for each cloth. And then after, we will stir for the 10 seconds for the 5, 10 and 15 minutes. And after 15 minutes, each piece of the cloth was removed and placed on the white tile which were labeled bio, non-bio, washed and not washed. And in the step number 7, the student observed the stain on each piece of the clothes. Now in this way, the observation of the stain is the source of the finding the results. So this is what the investigation. After that, we shall move toward the results, because this is alternative to practical. Examiner will give you calculated results. In these calculated results, the first is the bio, second is the non-bio, and water and the not washed. And if you see that in the key, the lowest intensity is represented with 1 plus, while highest intensity is represented with 4 pluses. The examiner can ask the questions, prepare the table to record the results, use the information in the figure 1.1 and in the table to match the intensity stain to the score for each piece of the cloth. So this is 3-mark question. So the marking scheme that include, first you're supposed to draw the table, which should consist of at least 2 columns and the suitable rows. And the second is the column with the heading for the second mark. And the third mark is the correct use of the scale to record the intensity score. Now we are moving toward the first, the table drawn with the internal line and the 2 columns. Now this is the table which consists of the 2 columns and the 4 internal lines for these the 4 variables, and 1 is for heading. Next mark is for the suitable heading. For example, the type of cloth in the first column and intensity score in the second column. And for the third mark, so the correct use of scale to record the intensity score. So this intensity means the lowest intensity is the bio. So we'll represent with the bio and then the non-bio and then the water and the not washed. So in this way, the lowest intensity is with the bio, we will represent with the 1 plus. And the intensity of the stain is left over with a non-bio, so that is with 2 pluses. And further with the water and not washed, highest intensity. And in this way, your 3-mark question table will complete. We are moving toward the further questions, and these questions also depend on the following information. For example, independent and dependent variables. Now, independent variables include that are under control of investigator, mean the investigator can set the values of these the independent variables, and they do not depend on other variables, and they represent along x-axis in the graph. For example, in this experiment, we have the type of the detergent, temperature, and type of stain, and volume of the water, and washing times. These all are the independent variables. So the examiner can set these values of independent variables, while the dependent variables include the result of your experiment and dependent on the value of independent variable, and they are represented always along y-axis in the graph. For example, the intensity of the stain is a dependent variable. We are moving toward the question. Now the question is the state the variable that was measured dependent variable in this investigations. So in this investigations, we measured the amount or intensity of the stain is a dependent variable. Now the second question state the one variable that was kept the same in this investigation. So it was the size and type and dryness of the clothes and the type of the food stain and not washed cloth. So they were kept same because they can affect our results. So anyone you can write for the 1-mark answer. And next is the question number 4. Identify one source of error in this step number 4 and explain how it might affect the results. So you're supposed to identify the error and explain the step number 4 that include one glass rod was used. Just remember when we were reading the investigation, so we underlined the word one glass rod. Now this one glass rod can be a source of the error. So this is a use of a same glass rod. Now explanation you can give, same glass rod can contaminate non-bio and water with bioenzymes. So because the contamination may possible, like the enzymes from the bio detergent can be transferred into the water and non-bio. So and that may affect your results. And X, you can also give the stirring times will be the different for each solution. So the soaking time will be the different, right? Because using the same glass rule. So any one point you can write about. Now the last question is like identify the control in this experiment and explain why this control was used. So the control is the any quantity in which we do not apply any treatment. For example, in this experiment, the treatment was the bio and non-bio detergents. And if you use the water, so that will consider as a control. Now the second mark is to explain why we use this control. So to compare the color of the control with the color intensity of the cloth with the different treatments. So the last questions we are heading toward another. The state why the method used in the step number 7 may not give accurate results. Now in this step number 7, the student observed the stain on each piece of cloth and they used the intensity score. Now the table 1.1 to determine the intensity of the stain on the 4 pieces of the clothes. Now the answer for these questions, so the qualitative judgment by the eye may not give accurate results because the different people have the different eyesight. So whenever the examiner will use the source of measurement is the color intensity or the different spectrums, so this can be a source of error. Now the stain on the underside of the cloth was not considered. Why? Because if we are observing one side of the cloth, so we can observe the only one dimensions. So in this way, like in the one direction, so we can observe the cloth. So it will consider another the source of error. So this is it, and that is the source of errors. So we have done. Thank you very much for watching this video. If you didn't understand any point or if you want to focus on any other point, so you can put in the comments. That will be more appreciable. Thank you very much.